chemistry, basic chemistry. A lot of you had that or are taking it. If you need more information about chemistry, I did put a little 10-minute video out on the canvas. If you need a refresher, watch that. It'll help you understand. I'm not going to ask you specific questions about it, but it will help you to understand some of the concepts that we're going to be talking about. And we started chapter 3 already, cells and methods of observing. We're going to continue that today and probably get into chapter 4 today as well. Okay, so it's hard to see bacteria because they're so small. So we have to do something to them to make them show up better. And we do that by so you can see in this picture that contrast stains, left side, right side. So stains are what we use to view microbes. But the stains kill the microbes. So the process of fixing the bacteria in this slide, which we'll get to in a little bit, kills them because they can eat. So if you want to study live organisms, you can, but you can't stain them. It's a little bit harder to see because of that. So let's have a look at these two pictures, left side and right side. Can anybody tell me anything about them just by looking at them? Thinking back to what we learned last week. What do you think? Okay, so tell me how you know that the left side is negative and the right side is positive. It's pink. Negative is pink, and so positive is purple. Anybody else remember that? Yeah. And there's also, it's hard to see because they are so small, but on the left side, the shape, what do we call that shape? It's hard to see. It's a rod. So what is that? Bacillus. And on the right side, the purple is a sphere. What's that? Top side? Yeah. Okay. So you know more than you think you know, right? So gram negative versus gram positive. We're going to talk more about this particular gram stain in a little bit. But we're going to talk about, let me go back, that's just types of microscopes. So there's all different types of microscope. We already looked at that light microscope, remember that? The bright field microscope, showed you a little video of that. You're going to use those in your micro lab. But there's other kinds of microscopes as well. There's a dark field microscope. It's exactly what it says. It has a dark field. So the field is the background. So a light or a bright field microscope, the background is bright. Dark field microscope, the background is dark. So it allows you to look at some things a little bit differently. This is an amoeba. An amoeba is a single-celled organism in the protozoa family, right? So if we're looking at amoebas, this is a good microscope to use because amoebas are clear, right? So you put this amoeba on a bright field microscope and you're not really going to see anything. So what we want to do whenever we are looking at organisms is increase the contrast. And contrast just makes darker things darker and brighter things brighter. Some of you take photos, you have Photoshop, you mess with the contrast, right, to make your pictures look a little bit better. Another kind of microscope is called the phase contrast microscope. This one, another amoeba. This one looks a little bit different though, doesn't it? Doesn't it kind of pop out a little bit more? It looks a little more detailed. Okay? And so it's just changing the contrast in a different way. So dense material looks darker. Less dense looks lighter. DIC microscopes. DIC is what we call them, but that stands for differential interference contrast. So again, we're talking about contrast, right? We're talking about making things more visible to our eyes by changing the brights and the darks. This amoeba, you can see it pretty good, can't you? It's pretty clear because you have a real three-dimensional image. You have the ability to see more of what's going on inside of that organism. It really does look like it pops out from the background. So all of these microscopes that I'm talking about, they just get more and more expensive, right? Yeah. The more they can do, the more expensive they are. So we've been looking at really the same type of organism, and you've seen how it changes the photographs as we get to microscopes that are able to give us more and more detail. A fluorescent microscope, just a little bit different. That word fluorescent, you probably already know what that means. Kind of a neon looking colors, right? Fluorescent means that light is affected differently by it. So we use these fluorescent dyes in some bacteria to make them show up better. But in order to use these dyes, we have to have the right kind of microscope to look at them. So a bright field microscope, you won't be able to see the fluorescent dyes. That one's pretty easy to remember. What kind of bacteria are these? What category? Shade?
It was like wow.
Bacillus? Did you say that?
Yeah.
Okay. Probably the most exciting microscopes are electron microscopes because they can magnify organisms from 100,000 times and some of them up to 10 million times the size. So the electron microscopes are the ones that enable us to see viruses. Viruses cannot be seen in any of the other microscopes that we just talked about. They're too small. So in order to see those, we have to use an electron microscope. There are a few different types of microscopes. It doesn't matter. Just know that the microscope that allows you to see the smallest organisms, or the microscope that magnifies the most, is the electron microscope. All of these use a computer because we can't see electrons. They're too small, and so we can't actually see. It's not like you looked at an eyepiece and see it. That's not what's happening. What happens is the electrons are projected onto the organism, and a computer renders a picture to us of what that organism looks like. So these are very expensive microscopes. I've never used one. There's probably 4 or 5 different types of electron microscopes. Again, they just get more and more expensive as they get when they can magnify more and more. This is a picture of one of the electron microscopes here at the University of Memphis. I don't know where it is, but I suspect it's near here because bioengineering. Maybe I can figure out a way to visit. But you see the microscope on the right side, and then the computer screen is showing you what it's looking at on the left side. And then if we were to just look at some images, when we think about, remember the slides I just showed you of the pink and the purple and just little dots, and you can hardly tell what they were at the very beginning of the class? Well, when you look at these are the same kind of organisms. We've got cocci, we've got bacilli, bacillus, coccus over there, and you can see how much more detail you get from these electron microscopes. So it's really, to me, amazing. You can see that they are absolutely three-dimensional, where they look so flat on the microscope slide. Okay, attendance. See, this is my sticky note. Just remind me. So we had a couple of quizzes. Most of you guys were able to get those quizzes done. If you need to communicate with me about a quiz, do so. I think I have a couple of new students, and you might not know what's going on, so just send me an email or come see me after class. We'll figure it out. You're going to have another quiz today. It opens up after class, it's 10 points, and it closes on Sunday night. All the quizzes are pretty much going to close on Sunday night, except quiz 2 that I gave you. I think I gave you a day extra on that one. You may even close today, because we got to get used to stuff, right? It's our first week of class, so let's get used to it. These quizzes are not only just easy points for you guys, you know how easy they are, you get chances to see the questions, to answer the questions, you get to see which ones you don't know, you get to study, find out answers to the ones you don't know, and you get to say it again. Okay? Same questions. So you need to do that. Plus, these quizzes give you an idea of the types of questions that will be on your exam. Some of the quiz questions will be word for word on your exam. So be sure you take these quizzes seriously. They add up to about 2 test scores by the end of the year. That could be 2 perfect scores you could get on 2 tests. So take the quizzes. All right. Okay, so when you get to lab, and you've had lab, but have you done a slide yet? Did you wash your hands? Yeah, that's good. Okay. So the things I'm going to tell you today, and a little bit probably on Thursday, are going to help you in your lab. Okay, so just sort of prepare you for what to expect in lab. So we're just going to talk a little bit about how you prepare a specimen. Some of you have done this, some of you did it in high school, some of you haven't done it yet. That's fine. It's all going to be fine. One of the simplest and easiest ways to prepare specimens is called a wet mount. And all of that is, you just put your specimen on a microscope slide, a drop of water, you put a cover slip on top of it. That's all that is, a wet mount. And we use this to observe living specimens. So if I want to see an amoeba, then I can use a wet mount. If I want to see that amoeba and how it moves around, or any other protozoa, any other protist. There is an organism. Oh, I should have put a picture in the slideshow for you. It's called a tardigrade, and a tardigrade is a microscopic organism that looks like an animal. It looks like a little bear, and it's called a water bear. And tardigrades are very durable. They're very difficult to kill. They've even been to outer space. So a tardigrade would be a living organism that you might want to look at under a wet mount so that you can see its motion and how it moves around. So in this picture, there's a couple of things we're looking at that is pointed out. We've got some bacteria and we have some yeast cells. So the yeast cells are big, okay? I know that looks like it could be maybe staph or some other spherical bacteria, but the spheres are all the yeast. In this case, they're circles or spheres. And the bacilli are really tiny and hard to see. So just kind of showing you yeast versus bacilli and the sizes. going to look at live specimens very often. What we want to do is we want to make a differential slide because it's going to tell us the difference between one cell and another. And so to do that, we have to do a smear. So wet mount looks at live organisms, but a smear is going to kill the organism. Oh, sorry. So the way we do a smear is we take a little bit of the specimen with a little loop, and that loop might be metal and reusable, and you would stick that loop in a flame and sterilize it, let it cool down, put it in your solution or your sample, and it fills up a teeny little loop. You ever blow bubbles as a child? And you know those little sticks you put in the bubble, the little round, you know, it gets a little bubble in there and you can see, yeah, it's just like that, but it's a lot smaller. So it's a little bit of film that gets in that little circle, and you just smear it on the slide, just like it says, and you want it to be thin. The biggest mistake that students make in lab is they don't have it thin enough because we can't see it. It looks like there's nothing there, right? It looks like nothing's on there. So we want to make it where we can see it. Don't do that. Just trust me, it's there, it's on there. If it's in the loop and you put it on your slide, it's there. You want it to be thin because you don't want to have a stack of cells. You can't see that very well. Okay, and then you're going to let it air dry. You're going to put it over a flame, and that's going to kill the organism. Then you're going to put some sort of dye. I'm going to talk about some different dyes in just a minute. And then you're going to look at it underneath the microscope. So wet mount is for live, and smears are for everything else. Okay. These are some of the stains you're going to see. You don't have to memorize that. I just want to show you what they look like. So they have a different purpose, a different function. They're different colors. Some of them adhere to cells in a different way. That's how you get the gram-negative and the gram-positive cells, depending on what the structure of the cell is. So simple stain is easy. Simple stain is one color. So this is very common. You make your smear and use one stain. Put it on there. You usually leave it for a few seconds, 15 seconds, 30 seconds, and then you wash it off just with water. The stain adheres to the organism, and then you're able to see it underneath the microscope. So you're going to be looking at these colorful things. You see on the left and right. Okay, so on the left side, there's a lot of big blobs. That's just stain. Okay. What you're looking at are the tiny little things, right? In the middle there. They're bacteria, and they're shaped like spheres. The right side, that's harder, isn't it? Look at that. There's a lot of stuff going on the right side. Yeah. So you know, it just takes practice. Don't get frustrated in lab when you look at something and it looks like the thing on the right side and you're like, I have no idea what this is. It's really okay. The more you look at it, the more you see it, and your lab instructors are going to help you with this, the more you'll be able to identify what you're looking at. And mainly over here on the right side, you're really looking at the cilia, but they are turned in different directions. Yeah. Okay. So you don't have to write this down. Just know that we have methylene blue, we have crystal violet. It's just the color and the name of the stain. Okay, but differential staining is a different process entirely. So a differential stain helps us to see the difference in the cells. That's what's called differential. Simple stain, one color, you look at it. But a differential stain, you're going to just go through a more involved process. And what you're trying to do is you're trying to see on your slide what kind of cells I might have, because they may not be all the same. Okay. So this is your gram stain. We covered this, but it's always good to review. We're trying to see the difference in that epitoplasm layer so we can identify if it's gram-negative or gram-positive. Ultimately, because if we know what it is, we can then figure out how to treat it, okay? Because we have drugs for gram-negative and we have drugs for gram-positive. And that's why this is so important. And you're gonna see this in lab reports. You're going to see a lab report come back and it's gonna say gram-negative bacilli, in the specimen. And so the physician is going to order the antibiotic for that. But you guys need to know this kind of stuff because you're the one giving that medicine. Yeah. Okay, so how do we do a gram stain? You'll get these instructions in the lab. You don't really have to memorize this, but I just want to go through and show you why we do it and what is happening when you are doing it. So you're going to first make a smear, okay? You're going to first make a smear, and then once you've got it fixed to the slide, you're going to do your staining process. So you've got crystal violet first, you put that on, it stains every single thing purple. Everything's purple. And then you're going to wash that off. You're going to put iodine on there. Well, everything's still purple. Okay, then you're going to wash that off with alcohol. Alcohol is a decolorizer. It means it takes some color out of it. Okay? And so what you're going to end up with is you're going to end up with purple for those gram positive, and then you're going to have gram negative. All the color's going to leave them. Okay? So the purple stains the gram positive balls, and the gram negative balls are colorless, and then you put an additional stain on there, and that's going to color those gram negatives. So that's how it works. So it's more than a one-step process. So here is just a quick little video.
Gram stain is a staining technique that classifies a bacterial species into one of two groups based on its color after the four-step staining process is complete. Gram-positive organisms will appear dark purple, while gram-negative organisms will appear red or pink. Begin with a properly prepared bacterial smear using a clothespin as a slide holder. Add crystal violet. Cover the smear completely and let sit for 1 minute. After 1 minute has elapsed, use a water bottle to rinse. Next, apply grams iodine and let sit for 1 minute. After 1 minute has elapsed, rinse the slide again. Next, add decolorizer until it runs clear. However, don't add decolorizer for more than 15 seconds. Rinse the slide again. Finally, add the counterstain saccharin and let sit for 1 minute. After 1 minute, rinse the slide again. Blot the slide dry with a piece of bibulous paper. The slide is now ready to be observed under a microscope. If prepared correctly, you should be able to distinguish gram-positive and gram-negative organisms.
Okay, so that was like fun, didn't it? And it is fun. rubbing off all your work that you just did possibly, right? Yeah, so you just pat it. Yeah, just carefully pat it dry. And then you'll be able to look at it and hopefully you'll see these organisms. You did your stain correctly. Okay, another kind of stain is called the acid-fast stain. I'm not sure if you'll do one of these or not, but you might. And in the acid-fast stain, there's a particular cell type called acid-fast cells, and they retain the red dye. Carbofuscin is what it's called. You don't have to know that. Just know that in an acid-fast stain, the cells stain red. And the big thing I want you to know is really this, because it's in red, right? So acid-fast stains identify mycobacterium. What is peculiar about mycobacterium? Mycobacterium has no cell wall, okay? So I can't use a gram stain on it, right? Because gram stains stain peptidoglycan. That's in a cell wall. If I have no cell wall, I can't use the gram stain on it, okay? So mycobacterium have to be identified using a different kind of stain, this acid-fast stain. And we can see them in the picture against the blue background. They stand out pretty good. You can see them pretty good. Does anybody remember about mycobacterium and which antibiotic would not work against them? Penicillin, right, yeah. So again, the reason why these organisms need to be identified is so that we can have the proper treatment. No cell wall, just keep that in mind. No cell wall in the mycobacterium. They're pretty prevalent. They cause a pneumonia that can make you pretty sick. Okay. What's the capsule? Y'all know. You have to only talk to me. So it does enclose the bacteria, and why? What does it do? The cat helps with attachment, it helps protect it. So in this picture you can see, this looks pretty odd, doesn't it? I mean, you know, you're looking at this, and I see what is going on here, maybe like little eyeballs or something.
Bless you.
But what we're looking at is the bacteria surrounded by its capsule. Y'all, anybody else get it? Okay, so not all bacteria have capsules, so this is another way that we can identify the type of bacteria that it is. Endospores. Hopefully you remember about endospores. So endospores are a dormant kind of bacteria. They are very difficult to destroy. They can be destroyed, but it's really difficult to do so. They might live for 100 years, maybe even more. There are particular bacteria that form these endospores. Tetanus is one of them, remember that? Yeah. So we're going to be talking about endospores again on how we can get rid of them. Because the endospore is we are going to begin chapter 4. Just how do these microbes grow? What conditions do they prefer? How do they divide? And then we'll talk Thursday on what we can do to control microbial growth. Okay. So prokaryotic cells divide by binary fission. And fission, the word fission just means splitting in half. That's what it means. Split it in half, fission. Don't get it confused with fusion. Okay? Fusion means joining together. So we have one cell that divides in half, and then we have two daughter cells. The two daughter cells are identical to the cell we began with. So this is a different kind of division than human cells or eukaryotic cells do. It's almost like cloning. You know the word cloning? Yeah. So you just take one cell, divides into, they are identical. When you have this kind of growth, it's called exponential growth. Exponential growth is when you have a bacteria that population that doubles in a particular amount of time, and that time is called a generation time. So for instance, Streptococcus has a generation time of about 20 minutes. Okay? So if you get infected with strep throat, you've had that before, the sore throat, bacterial sore throat, very common, especially common in kids. So if you get strep throat, let's say that your initial dose is 100 strep bacteria. In 20 minutes, you have 200. 20 minutes more, 400. 20 minutes more, 800. 20 minutes more, 1600. Okay? So it doesn't take very long for you to have an enormous amount of strep bacteria in your throat. Just a few hours. And that's why it can kind of come on suddenly if you've ever had strep throat. It's very different from a cold. So a cold, you get that little scratchy feeling and nagging, you know, but it's not really sore. Strep throat is very different from that. You have nothing and then all of a sudden you have fever and sore throat. Yeah. Because of this exponential growth along with a short generation time. Okay. So, answer the question. If the culture starts with 50 cells, how many cells will be present after 5 generations? And we're always going to just assume in these problems that none of them die. They all are alive. Start with 50. How many will you have after 5 generations? You can talk to a neighbor if you want to. Check with a friend. You may know it already. That's your really good mouse. I hear 800. 1600. Anybody else? Would you agree? Don't know? Okay, here's how you do it. So this is just how I just make a picture of it because, you know, I like to see things in pictures. So 0 is your start time. 0 is what you begin with. I began with 50 at 0. And then I've got 5 generations, right? And each generation is doubled. So 1600, thank you. 1600 is correct. Okay? There's a formula you can use, but yeah, you don't have to do that. You just count it up. It's not that hard. Be able to do 1 or 2 of these. It's not hard. You just have to know how many times to double it, right? That's all you have to know. How many times do I double it? And cells don't just multiply as they're flowing through the bloodstream. They attach to something. And sometimes they use their capsules to attach. Remember, we talked about those. Sometimes they have other little pili or other little structures on their surfaces that help them to attach. And when they attach, they secrete some extra polymeric substances, the EPS, and that's what gives biofilms a sort of slimy appearance. Have you ever found something stuck in the back of your fridge in a vacuum? And you pull it out and it's a slimy mess? Yeah, because it was once, green pepper, and now it's a slimy mess. That's a biofilm. Yeah. A biofilm that we can see. These bacterial cells communicate with each other. We will talk much more about cellular communication later on. So just know that cells communicate with each other with chemical signals. They can communicate with each other, the cells right in their vicinity, and they can communicate with cells that are a little bit further away with these chemical signals. They can attract cells to them, and they can get to a point where they begin to replicate or divide frequently through something called fluorescence. So just know that it's not random. These bacteria have a method in mind. So let's have a look at just how a biofilm might form. We've got, this is some bacteria that adhere to a surface. You have that EPS secreted. And you can get other kinds of bacteria too. It's not always the same species. Okay? See, it's just a collection of bacteria that begin to grow and grow. And sometimes you get bacteria that break off and go elsewhere and form another kind of biofilm. So biofilms. Biofilms are found in your body, and biofilms are found outside the body. Sometimes you find them in shower heads, growing on the walls of showers. If you've ever seen pink stuff growing in a public shower, hopefully not your own home shower, that's a biofilm. Okay. So if we looked at a biofilm up close, this is what it looks like. It's a collection of bacteria and other substances. So imagine if you got this sort of thing in a blood vessel of your heart. Can you imagine that? Yeah. So you can see how a biofilm in a blood vessel could clog it up, right? Yeah. Okay, so biofilms. Let's just talk about some of the health-related concerns. Plaque, dental plaque. So, brushing your teeth, flossing, these kinds of things remove the plaque every day. So this plaque grows in your mouth. It takes about 24 hours kind of to set up. That's why you should brush your teeth and floss every day. Okay? Dental plaque can lead to tooth decay. It can lead to gum damage. Gum damage can lead to your teeth falling out. So it can be bad for your oral health. And we will talk more about this when we get to some of the diseases of the body. Sometimes plaque can be mobilized in the bloodstream and go to your heart blood vessels. This can really happen. Some heart attacks and cardiac disease are caused by the bacteria in the mouth. So we are just learning more and more about this read through it. So this is kind of like thinking through something. Let me get back here so I can read it better. Certain types of bacteria, Streptococcus mutans, thrive in the innermost anaerobic layers of the plaque. Anaerobic, we haven't had that word yet, anaerobic. What does it mean? Who knows? Anaerobic. No oxygen. You're Alex, right? Okay, Alex says no oxygen. That's what it means. An means without or no or none. Aerobic means oxygen. So anaerobic. Layers of the plaque and are associated with cavities, which produce acids. Other types of bacteria, Streptococcus sanguinus. So they're both Streptococcus, but they're different kinds of Streptococcus. Compete with S mutants, but are unable to thrive in that acidic environment. Normal toothbrushing effectively removes much of the plaque from the flat surfaces of the teeth, but cannot reach the surfaces between the teeth. Most commercial toothpastes contain alkaline, that's basic, components which raise the pH of the mouth. Acid lowers the pH of the mouth, okay? So, all that information, can you predict? How is the population sizes of S mutants and S sanguinus in the bacterial community in the plaque between the teeth? How are they likely to change when these toothpastes are used? Okay. So go back up, figure this out, talk to somebody next to you or behind you or in front of you, that's fine. I want you to figure out, how does toothpaste, the alkaline toothpaste, affect the population sizes of these two different kinds of bacteria? You can talk to somebody, that's fine. It's okay to talk to each other. Basically, you have to figure out which kind of bacteria is going to be affected by alkaline and which kind, that's really what you have to figure out. So, which one's going to grow or which one's going to not grow in the presence of this alkaline environment? That's what you're trying to figure out. Some of you think you have it? I know it's tough. Let's see. Did you think this S mutans going to decrease, S sanguinus is going to increase? How do we figure that out? Look up there at the top. Mutants thrive in areas associated with acid. You see that? So mutants thrives in acid. If I use an alkaline toothpaste, it changes the environment, less acid. Therefore, that one's going to decrease. Sanguinus increases with alkaline. It says that up there. And so it's going to increase. So you have hundreds, probably thousands, really, of different kinds of bacteria in your mouth. And that just kind of puts a whole new focus on kissing, doesn't it? And those bacteria can cause cavities. And some dentists they've been around forever, they know what's going on. Some people have cavities after cavities. Some people have never had a cavity in their life. Why? Because of the kind of bacteria growing in their mouth. That's why. So if you've never had a cavity in your life and don't have a filling in your mouth, you're very lucky. You have the kind of bacteria that doesn't cause cavities. You're not like me with a bunch of fillings in your mouth. Okay. So in the lab, we we are going to approach microbial growth a little bit differently. Because in a lab, don't we want microbial growth? Yeah. So in our labs, we want stuff to grow so that we can see and do experiments on it. We can see what would kill it. We can see what would decrease its growth or increase its growth. So let's talk about the lab. So in a lab, sometimes we want pure culture. In fact, that's really kind of our goal most of the time. We want pure culture, where we only have one kind of bacteria that divides from one particular cell. So that's our goal. This is hard to reach because we have a whole bunch of different kinds of microorganisms in the environment, right? And sometimes we contaminate our sample because we weren't careful enough. So you're going to learn some of the techniques on how to have how to collect a good sample, how to keep that sample as pure as possible. And the thing you're going to be focusing on is called aseptic technique. Sometimes this is called sterile technique. So in the medical profession, aseptic technique is really important, especially when you're doing a procedure where you're putting something in. And you're going to be doing those procedures. For instance, you're going to give some injections. Okay, we call those shots, right? You're going to give shots, and those injections need to be done in a sterile, aseptic manner. You may start. You will start IVs. IVs are little plastic tubes, we call them catheters, that go into a vein. You're going to do that. You want to be sure you have aseptic technique for that, right? So the things you learn in lab that are sterile technique or aseptic technique, keep those in mind because you're going to be using that. And we'll talk more about this when we talk about procedures. Okay. In the lab, though, we're going to use aseptic technique. We're going to be sure that we don't contaminate our sample. With those little loops, we're either going to plane those loops, or you may have disposable loops. I'm not sure what you use in your lab. Disposable loops have already been sterilized, and they come in a package. And when you take that loop out of that package, if you lay it down on your lab bench, it's contaminated, right? Yeah, so don't do that. If you touch the end of it, the little circle end, it's contaminated. So you have to learn how to take that thing out. Don't touch it on anything except your sample. Yeah. And then sometimes you can't reuse it. You have to throw that one away and get another one. Or if you're using a reusable metal wire loop, you have to sterilize it in a flame again between each sample. Okay? So just keep in mind, those are kind of the sterile techniques you're going to learn. And then you're going to grow your cells in something called a culture medium. And the culture medium is just the nutrition that the cells need in order to grow and divide. Now, you may also do lab where you have a particular organism and you put it in different kinds of growth to see what it grows on and what it doesn't grow on. So there's different kinds of nutrient mediums that you might use. We're not going to really talk about some of those specific kind of mediums because you'll learn that in your lab as you do your particular experiments. So you might have organisms that you're growing in a liquid, or you might have organisms that you grow on something that looks more solid. It's not solid like this table though. You could dig it out, gouge it out with your loop. So you have to be kind of careful on that. It's thicker than Jell-O, not that, loose. I'm trying to think of what it would be like. I can't even think of anything. Maybe like cheese. Maybe like a little bit of maybe a softer kind of cheese perhaps. You'll see what it's like. So, to obtain a pure culture. This is your lab directive. You've been told to obtain a pure culture. So you're going to need a few things. You're going to need the medium. What's the medium? What's going to grow on, right? Okay. You're going to need some kind of container. Usually we use petri dishes. We'll talk about that in a minute. And you're going to need your aseptic conditions. If you contaminate your sample, then that sample is useless. Okay? So don't contaminate your sample. I guess what I'm telling you is you're going to get instructions on how to do the procedure. Just read carefully and follow those instructions carefully. Yeah. And you'll be successful. So you're not going to see a single cell. You're not even going to see 100 cells. You can't see those cells until a colony gets to be about 1,000,000 cells. Okay? That's how small bacteria are. And that's going to be your goal. You're going to take something you can't see, put it on the growth medium, incubate it, let it grow a little bit until you can see it. And sometimes you'll do something called a colony count, where you count the number of little areas that are growing, and that gives you an estimate of the number of cells there based on 1,000,000 cells easily seen. So it's just a little way that you can count it, give you an estimate of the growth. So maybe your experiment is, I have an E. coli that's safe. There's safe species in E. coli that doesn't infect, non-infectious pathogen. And I want to see what kind of medium it grows best on. So I have 3 different mediums, and I put my sample on each one, and then I come back after it's incubated and after it's had its grow. You know, it's gone through its generation time, it's gone through the binary fission, and all these things we just talked about. And then I can come back and I can see my colonies, and I can see which medium it grew best on. Agar is what we use to solidify a medium. Your lab techs will probably do this for you. You won't be doing this yourself. So you'll already have your medium, your culture medium made for you. On the left side, we have some bacteria, cells, and nutrient agar that are in little test tubes, just kind of showing you what you might see. And on the right side, we have more of a thin broth in a flask. And you might have either one of those, whatever you've been given. So on agar, when it says few microbes can degrade, it means that they don't die because it's a good medium for them to survive on. That's all that means. Agar is also really great because it can be sterilized. So it's not degraded or melted by the high temperatures. It can be sterilized so that you can have a fresh, non-infected culture medium. So the way you're going to do this is you'll be given a test tube or a flask or some other little container. Inside of that, sometimes it's a little bitty bottle, you know. It just depends on what your lab has at the time. You'll put your loop in there, your sterile loop, right? Not anything that you've laid down on the counter, because now if you put that in there, you've contaminated your whole source. You'll put your sterile loop in there, take it out. Don't set it down until you put that organism on your agar. We're going to talk about how you do that as well. And even though we can heat this and melt it down, it'll solidify back up. And the temperature range for most microbial growth is, we'll get to that in a minute, but just so you know, it's about body temperature. That's why they like our bodies so much, because we are the perfect temperature for some of these pathogens. Okay, so this is your petri dish. If you've never seen one of these before, it's a really little thin round container. You can see how big it is. It's about the big as your palm of your hand, and it has a little cover on it. And so when you get a petri dish, it's going to be covered. Okay. So as you start to get your sample ready, you don't want to uncover your petri dish and just leave it there while you're doing your stuff. Why? There are microbes in the air. There are. There's yeast and there's fungus in the air. We're breathing them in right now. And that could contaminate, could fall on your agar, your growth medium, and contaminate it. And you don't want that. So keep your petri dish covered until right before you need to use it. So you're going to take your lube, you're going to sterilize it or use a sterile lube. You're going to put it in your microbial sample. It's going to fill up the little bubble with a little film. And then you're going to go over to your petri dish with the agar on it and open the top. You don't even take the top off. You don't even take it all the way up. Just lift it up on one side. Stick your loop in there, okay? And that's how you're going to do it. And then you're going to put the top back on. And that's all aseptic technique. It's not hard. It's honestly kind of fun, okay? So these petri dishes, though, do allow, when I say air, oxygen, they do allow oxygen to enter, but not contaminants. So the way they're formed, the lid sits on top. It's not like screwed on, it just sits on top. And so because of that, oxygen is allowed to get in there. Because some microorganisms are aerobic and they need that oxygen. In fact, what you're going to be working with are aerobic. And then an agar plate is simply a petri dish with agar medium in it. So you'll hear that term. Put this on your agar plate, and you'll be going, I don't have a plate here. Well, it's talking about the petri dish with the medium on it. So that term is just agar plate. And that is not liquid, it's a solid substance. It's a soft solid. Okay, so let's just sort of go through some steps of how we would streak our plate. That's what it's called. We're making a streak plate. What are we doing this? So this is really the most common method of isolating or growing cells. Sorry about that. Step number one, you're going to sterilize this, or you're going to use a sterile loop. Step number two, you're going to put that sterile loop into your culture. In this case, it's a test tube. Step number three, you're going to streak part of the plate. Do you see that? Just part of it. All that's pretty straightforward. But what you're going to do next is what you're trying to do is reduce the number of bacteria. So you can get down to just a little bit of bacteria so you can get the growth that you can actually count. So then you're going to sterilize your loop again or get a new loop. But you don't dip it back in the culture. You're going to actually use the plate, what you just streaked. You can see that you streaked it. You're going to use that, and you're just going to drag it over to another area. Then you're going to do that again and drag it over to another area. So you're diluting your sample, okay? You put it down, and then you kind of smear it again, and then you smear it again, but you're smearing it into smaller and smaller and smaller amounts of that. Also, when you put that lube on your agar, barely just touch it. Don't dig down into it. It's soft. So kind of hold it at an angle and just barely, go over it. So what you're trying to do is reduce the number of cells that you have. If you just put it on there once, you're going to get so much microbial growth you can't count it. So you just do the streak plate method so that you can reduce the number of cells as you go along. Now, in this case, they've got this opened up. I prefer to do it just like we talked about. You've got your Petri dish, it's got a little cover on it, it's sitting on the table. You lift up the cover and you streak, and then you put the cover back in. And then you either clean your loop or get a new loop, whatever's happening in your particular case. And then when you go back, you kind of turn the plate, and then you just, lift it up and streak and put it back down. And that's going to avoid contamination. You can do it, it's not hard. So ultimately, on the left side is the drawing of what we just talked about, and on the right side is the growth. And see what you're trying to do? You're trying to get those isolated colonies, those little round circles. So let's watch a little video on how to do it.
To streak a plate. To streak a plate, you'll need the bacterial cells you're plating, a fresh agar plate, and something to spread with. Perhaps a sterile toothpick, pipette tip, or wire loop. If you're using a wire loop, you'll also need a Bunsen burner. Start by gently touching your stock cells with the toothpick. Just a soft touch is enough to get all the cells you'll need. Carefully, using a back and forth zigzag motion, spread the cells across one side of the agar. Don't apply too much pressure, or you'll damage the agar. This first area will form a thicker lawn of cells. To produce individual colonies, grab a fresh, sterile toothpick and drag it through the area you just spread cells over. Use that same zigzag motion to spread them out even more. But wait, we're not done yet. Grab one last sterile toothpick and repeat the same motion. Drag it through the second area and spread the cells in a zigzag motion. This third area will give you the best chance at isolating single colonies. If you're using a wire loop, the process will be exactly the same, except you need to sterilize the loop in between streaks. Hold the wire loop in the flame of the burner for 2 to 3 seconds, then be sure to let it cool before touching the cells. Place your plate in the correct incubator. Tomorrow, you'll be able to see single colonies. This video is brought to you by JM109 Competent Cell.
Thank you so much for bringing us that video. Okay, so they did not put the cover on it because they were videoing it and they wanted you to be able to see it. But use that cover to keep everything going. Okay. It's gonna be fun when you do that. So as you know, microorganisms are everywhere. You already know that. We haven't really talked about extremophiles much. Do you remember the 2 domains? 2 domains of cells. Prokaryote, eukaryote, remember that? And then the two divisions under the prokaryote. Bacteria and archaea, remember that? Okay. So extremophiles are usually archaea. They are prokaryotic cells. They don't have a membrane-bound nucleus. They are honestly almost like bacteria. Okay, but they know that archaea can live in extreme situations of these factors. So temperature. So when I say temperature extreme, I mean really, really high or really, really low. Atmosphere, that's pressure. High pressure, low pressure. pH, high pH, low pH. Water availability. Lots of water, not any water at all. So extremophiles can live in those edges where bacteria generally grow right in the middle. So, if we know what factors affect microbial growth, we can manipulate those factors. And that manipulation will affect that microbial growth, right? Yeah. So, if we want to kill a bacteria, we can heat it up, right? That's why you should cook your meat to 165 degrees. Because 165 degrees will kill all the pathogens that might be in your hamburger. So that's kind of like why we do what we do, right? Okay. So we're just going to look at oxygen real quick and just talk about some of those terms. And Alex already told us about anaerobic, and we talked about aerobic. So there are some kinds of microorganisms that are obligate aerobes. Obligate means I have to have it. I cannot live in any other condition. Okay? So an obligate aerobe is a bacteria that must have oxygen. But a facultative aerobe, or anaerobe, is one that can grow in oxygen. It prefers oxygen, but it can grow without it. Okay? So there are some bacteria that grow best in oxygen, but they can grow without it too. So taking away their oxygen supply would not get rid of it. Obligate anaerobes, I only grow without oxygen. If you expose me to oxygen, I will not grow. And then microphilia, those last two, they're very rare. We don't really see those much. The ones we're most concerned about are the obligate aerobe, obligate anaerobe, or the facultative anaerobe. Those are the bacteria that are most common. Okay. So aerobic uses oxygen, anaerobic do not use oxygen. I don't know if you've ever heard of aerobic exercise. That was a big deal. They actually called it that before when exercise was getting really, really big, like group exercise, like in the 80s, everybody did stuff. And they did aerobic exercise. Because as you do exercise, you require more oxygen because it makes you breathe more, right? Yeah. And you're using more calories and you need more oxygen for all of that reduction. Anaerobic would not use oxygen. All right, pH. Hopefully you know what pH is. What is it? pH. You don't have to know what pH stands for. But when I say pH, what comes to mind? Acidic, alkaline, acid, bases, right? That's what comes to mind. Yeah. So that's all that is. So we have acid, neutral, base, right? Yeah. And if you remember, there's a scale of 1 to 7, 1 to 14. 7's neutral. It's coming back. Under 7's an acid, above 7's a base. Bases actually feel kind of slimy, like chlorine bleach is a base. Have you ever felt chlorine bleach? It's got a little slimy feel to it. Acids don't have that, like lemon juice. That is, that's not slimy at all. Yeah. So, acids and bases. So the range for most microorganisms is 5 to 8. So microorganisms want to grow in a pH of 5 to 8. That's their healthy range. And 7 is really perfect. And guess what what pH is? 7. Yeah. So we are an environment, our pH environment, I should say, is pretty optimal. Now, we have one area of our body that has a really low pH, like 3, 2, what is it? Stomach acid, that's right. So our stomach acid is not conducive to bacteria. And in fact, a lot of times we eat something, we bring, I mean, our hands aren't clean, we put that sandwich on the table, you know what I'm saying? We put stuff in our bodies that have microorganisms on them, but our stomach acid kills those. Okay? It's one of our lines of defense. Now there is something that lives in stomach acid, we've talked about it, H. pylori. So there's always that exception to the rule. So neurophiles, I should say, are microorganisms that live in the human body at about 5 to 8. 7 is their perfect or their optimal. Acidophiles grow below 5.5. H. pylori is one of those that grows in our stomach acid. And alkalophiles are going to grow above 8. Most infectious bacteria are neutrophils, but there are exceptions. And then there are archaea that can live at either extreme and are perfectly happy. So, oxygen affects microbial growth, pH affects microbial growth, water availability affects microbial growth. All I really want you to know is all microorganisms require water. Now, if water is taken away, some microorganisms become endospores. Dormant cells, just waiting to be reactivated. We're not going to talk about concentration and pulling it out of the cell. Just know water is important for microbial growth. Okay. What is this? Let's not talk about that. Let's. I'm not sure what it was. I think my slides are out of order. All right. I tell you what, we're going to talk about, I want to talk about temperature. We'll talk about it on Thursday. Let us just go along. Until your next class. That's a lot today.
The signage?
All right,